500 million years ago, the oceans of Earth bore no resemblance to the waters we know today. There were no whales singing in the depths, no schools of fish flashing silver in the sunlight, no seabirds wheeling overhead. The land itself lay barren, a rocky wasteland untouched by plant or tree. Yet beneath these ancient waves, something extraordinary was beginning. In the silent darkness of the Cambrian seas, the first architects of living stone were discovering an alchemy that would transform the planet forever. Before we continue this journey, tell us where you're tuning in from, and if this story resonates with you, make sure you're subscribed, because tomorrow I've saved something extraordinary for you. These pioneer builders were not corals at all. They were strange, vase-shaped organisms called archaeocyathids, creatures so alien to our modern understanding that scientists still debate precisely what they were. Imagine a cup made of perforated limestone, no larger than your thumb, attached to the seafloor by a holdfast that gripped the ancient rock. Within the double walls of these tiny cups, water flowed through hundreds of microscopic pores, filtering nutrients from the Cambrian currents. Each individual archaeocyatid was unremarkable, but together they discovered something profound. They could build. The process began simply enough. These organisms extracted calcium and carbonate ions from the seawater flowing through their porous walls. Through biochemistry, we are only beginning to understand. They combined these dissolved minerals into solid calcium carbonate, limestone, depositing it grain by grain around their living tissue. One archaeocyatid building its limestone cup was merely interesting, but when thousands grew together, when they built upon the foundations of their ancestors, when generation after generation added their contribution to the structure, they created Earth's first reefs. These reefs rose from the Cambrian seafloor like underwater cities, some stretching for kilometers, reaching heights of 30 meters or more. They were the first three-dimensional habitats in Earth's oceans, the first places where life could live not just on the bottom, but up in the water column, at different heights. In different neighborhoods of the same structure, the archaeocyathids had invented apartment living for marine creatures. Around and within their limestone towers, other organisms found shelter. Trilobites scuttled through the passages. Strange worms tunneled into the rock. The reefs became oases of abundance in relatively barren seas. For 30 million years, these reef builders dominated shallow tropical waters across the planet. Their constructions spread across what would become Antarctica, Australia, North America, and Asia. Then, approximately 490 million years ago, they vanished. The archaeocyathids disappeared from the fossil record almost completely, their extinction as mysterious as their origin. The reefs they had built remained as limestone monuments, but the living architects were gone. The oceans returned to a flatter world. Their first experiment with reef building apparently ended. But life does not abandon successful strategies easily. The genetic blueprints for extracting minerals from seawater and building solid structures had been proven. The ecological advantages of creating three-dimensional habitats had been demonstrated. Nature would try again, and when it did, it would create something even more spectacular. 50 million years after the archaeocyathids vanished, new reef builders emerged. These were the true corals, though they looked nothing like the branching staghorns or rounded brain corals we know today. The tabulate and rugose corals appeared in the Ordovician period, some 440 million years ago, and they would dominate reef building for the next 200 million years. Consider that span of time for a moment. 200 million years. Modern humans have existed for perhaps 300,000 years. These coral empires persisted for nearly 700 times longer than our entire species has walked the earth. The tabulate corals grew in sheets and chains, their polyps connected by horizontal platforms of limestone. If you could have swum through an Ordovician reef, running your hand along its surface, you would have felt like you were touching a vast honeycomb, each hexagonal chamber holding a tiny polyp. Thousands of them joined in a colonial network. The rugose corals took a different approach. Solitary or forming small clusters, they built conical or horn-shaped skeletons, earning them the nickname horn corals. Some grew to the size of a human forearm, massive solitary polyps that looked like inverted ice cream cones carved from stone. These Paleozoic reefs were enormous. In what is now the Guadalupe Mountains of Texas and New Mexico, you can walk through the remains of the Capitan Reef, a limestone barrier that once stretched for hundreds of kilometers along the edge of an ancient sea. When you stand at the base of El Capitan Peak, you are standing at what was once the crest of a coral reef, a structure built over millions of years by countless trillions of polyps, each adding its microscopic contribution to the whole. The scale is humbling. The peak rises nearly 1,000 meters above the desert floor, all of it reef. Through the Silurian and Devonian periods, as the first plants colonized the land and the first forests began to grow, coral reefs flourished in tropical seas. They were different from modern reefs in many ways. The water was different, the chemistry altered by different atmospheric conditions. The creatures that lived among the corals were different, bizarre by our standards. But the fundamental pattern was the same. Tiny polyps, extracting minerals from water, building limestone structures that created habitat for countless other species, transforming flat seafloors into vertical worlds of astonishing complexity. Then came the apocalypse. 252 million years ago, at the boundary between the Permian and Triassic periods, Earth experienced the most severe extinction event in its history. We call it the Great Dying, and the name is not hyperbole. 96% of all marine species disappeared. The oceans became graveyards. Volcanic eruptions in what is now Siberia released massive quantities of carbon dioxide and other gases, triggering a cascade of environmental catastrophes. Ocean temperatures soared, the water became acidic, oxygen levels plummeted. In some places, the seas became toxic stews where nothing could survive. The tabulate and rugose corals, after their 200 million year reign as Earth's master reef builders, were wiped out completely. Not a single species survived. The reefs they had built remained as limestone tombstones, but the living architects were extinct. For millions of years afterward, there were no significant reefs on Earth. The great three-dimensional cities of the ocean were gone, reduced to rubble and fossil beds. Yet even in this near-total destruction, a few coral survivors carried forward the genetic blueprints. These were the Scleractinian corals, a group that had appeared late in the Permian, just before the catastrophe. They were unremarkable at the time, minor players in reef ecosystems dominated by their tabulate and rugose cousins. But they possessed something their relatives lacked. They survived. The Scleractinians that made it through the Great Dying were different from their Paleozoic predecessors in crucial ways. Their limestone skeletons were built differently, constructed from a mineral form called aragonite rather than the calcite favoured by earlier corals. This might seem like a trivial detail, a footnote in mineralogy, but it would prove significant. Aragonite allows corals to build faster, to repair damage more quickly, to respond to environmental changes with greater flexibility. When the world was ending, these small differences meant everything. For tens of millions of years during the Triassic period, these survivor corals slowly recovered, rebuilding reef ecosystems with painful gradualness. Then, approximately 240 million years ago, they made a discovery that would change everything. They formed a partnership. The partners were microscopic algae, single-celled organisms called dinoflagellates that could photosynthesize, turning sunlight into energy. Somehow, through a process we still do not fully understand, these algae began living inside coral tissue. It was a strange arrangement, but it worked. The algae received protection and nutrients from the coral. In return, they shared the sugars they created through photosynthesis. This energy subsidy was revolutionary. Corals with algae partners could grow faster, calcify more rapidly, build larger structures. They could thrive in nutrient-poor tropical waters where unpartnered corals struggled. This symbiotic relationship, this ancient handshake between animal and plant, would eventually become so successful that we now call these symbiotic algae zooxanthellae, and they live inside virtually every coral in every modern tropical reef. The partnership painted the ocean with color. The algae gave corals shades of brown, green, blue, purple, and gold. Reefs transformed from grey limestone structures into living rainbows. But the evolution of modern reefs was not a straightforward progression. Earth experienced several more mass extinctions, each leaving its mark on coral evolution. The end of the Triassic, 201 million years ago, killed off many of the early recovering reefs. The end of the Cretaceous, 66 million years ago, the same catastrophe that ended the dinosaurs, devastated reef ecosystems again. After each extinction, corals recovered, but they came back different, having learned new survival tricks, having evolved new strategies. Consider what these repeated catastrophes taught them. Corals learned to expel their zooxanthellae partners when conditions became too stressful, a desperate strategy that gave them a chance to survive short-term heat waves even at the cost of losing their primary energy source. They learned to broadcast billions of eggs into open water, ensuring that even if local reefs were destroyed, their offspring might find distant shores. They developed the ability to reproduce both sexually and asexually, hedging their bets against catastrophe. Each extinction was a filter, removing the unlucky and the inflexible, allowing only the most resilient to continue. The world's geography itself shaped coral evolution. Through deep time, the continents have wandered across the globe, carried by the inexorable movement of plate tectonics. Reefs that formed in tropical waters found themselves pushed toward the poles as continents drifted. The Tethys Sea, which once separated the northern and southern continents, became a coral highway connecting oceans, allowing species to migrate vast distances. When this ancient seaway eventually closed, giving rise to the Mediterranean, populations were isolated, forced to evolve independently. Approximately 50 million years ago, as the modern world began to take shape, coral reefs occupied much of the tropical ocean. The partnership with zooxanthellae was well established. The basic body plan of modern corals was set. The Great Barrier Reef began forming perhaps 20 million years ago, as Australia drifted northward into tropical waters. The Caribbean reefs developed their own unique communities of species. In the Indo-Pacific, the Coral Triangle region emerged as the global center of coral diversity, a biodiversity hotspot that would eventually harbor 3/4 of all coral species. Throughout this vast sweep of evolutionary time, coral reefs have been both resilient and fragile. They have survived extinction events that erased most life on Earth. Yet they have also been wiped out repeatedly, their recovery taking millions of years. They are ancient and modern, primitive and sophisticated, simple in their basic biology, yet capable of creating the most complex ecosystems on the planet. When you look at a coral today, when you see a brain coral perhaps 300 years old, its surface a maze of ridges and valleys carved by the growth of its polyps, you are looking at the end point of 500 million years of evolution. You are seeing the inheritors of the archaeocyathids, who first turned dissolved minerals into living stone. You are witnessing organisms that have survived everything Earth could throw at them, every extinction event, every climate change, every catastrophe. The corals building reefs today carry within them genetic wisdom accumulated across 500 million years. They are not simply animals. They are living records of Earth's history, monuments to the power of life to persist, to adapt, to rebuild after every catastrophe. The reefs they construct are not just beautiful. They are testaments to resilience written in limestone, proof that even after the world ends, life finds a way to begin again. And now, in our modern age, these ancient survivors face a new challenge, one that comes not from volcanoes or asteroids or natural catastrophe, but from us, from the carbon we release into the atmosphere, from the warmth we add to the oceans, from the changes we bring to the chemistry of the seas. The question that confronts us is not whether corals can survive. They have proven that across deep time. The question is whether we will give them the chance to do so again. Tomorrow, we will dive deeper into the extraordinary chemistry that allows these organisms to build, to grow, to create structures that can last 10,000 years. We will enter the microscopic realm where the real magic happens, where calcium ions flow and crystals form, where the partnership between animal and algae operates with a precision that surpasses our finest technology. For now, remember this. The coral polyp you might see while snorkeling, barely the size of a pinhead, is not just a simple creature. It is the product of evolution older than trees, older than flowers, older than any animal that has ever walked on land. It carries within its cells the accumulated knowledge of 500 million years of reef building, and it is still, even now, engaged in the same ancient work that its ancestors began in the Cambrian seas, turning dissolved minerals into living stone, building paradise beneath the waves. To understand how corals build their limestone kingdoms, we must journey into a realm invisible to the naked eye, into a world of molecules and ions, of chemical reactions so precisely orchestrated that human technology cannot yet replicate them. Inside each coral polyp, no larger than a grain of rice, operates a chemical factory of staggering sophistication. Every second of every day, for as long as that polyp lives, this factory transforms dissolved minerals in seawater into solid stone. The raw materials arrive continuously. Seawater contains calcium ions, positively charged atoms of calcium floating freely among the water molecules. It also contains carbonate ions, negatively charged molecules made of carbon and oxygen. These ions exist in relatively low concentrations, about 400 parts per million for calcium, even less for carbonate. Yet from this dilute solution, corals extract what they need with remarkable efficiency. The process begins in specialized cells at the base of the polyp, in the thin layer of tissue that sits directly on top of the coral skeleton. These cells, called calicoblastic cells, are unlike any others in the animal kingdom. They possess the extraordinary ability to pump calcium ions from seawater, concentrating them in the space between the living tissue and the skeleton below. Imagine a cell that can reach out into the surrounding water, grab specific atoms, and pull them inward against the natural tendency of those atoms to disperse. This requires energy, substantial energy, and the cell burns through it constantly, maintaining concentration gradients that defy the laws of diffusion. Simultaneously, other cells are working to concentrate carbonate ions in this same space. But carbonate presents a problem. It exists in seawater in several forms, in equilibrium with bicarbonate ions and dissolved carbon dioxide. The balance between these forms depends critically on pH, on the acidity or alkalinity of the water. Corals need carbonate ions specifically, not bicarbonate or carbon dioxide. So they manipulate the pH in the calcification space, making it more alkaline, shifting the chemical equilibrium toward the carbonate form they require. When calcium ions and carbonate ions meet in sufficient concentration, they combine spontaneously to form calcium carbonate. But here is where the true sophistication becomes apparent. Calcium carbonate can crystallize in several different mineral forms, each with its own crystal structure, with its own properties. Calcite is one form, the type that makes up limestone and marble. Aragonite is another, slightly less stable but faster growing. Corals produce aragonite almost exclusively, and they do not produce it randomly. They grow it in precise patterns. The skeleton of a coral is not a blob of limestone haphazardly deposited. It is an intricate architecture of crystals, arranged in specific orientations, forming structures called sclerodermites, radiating bundles of aragonite needles that grow outward from centers of calcification. Under a microscope, a cross-section of coral skeleton looks like a starburst frozen in stone, each needle precisely aligned, each crystal face oriented according to genetic instructions encoded in the coral's DNA. How the coral controls this crystallization at the molecular level remains one of the great mysteries of marine biology. We know it involves organic molecules, proteins, and sugars that the coral secretes into the calcification space. These molecules act as templates, as guides that direct where crystals form and how they grow. But the details, the precise mechanisms, still elude us. The rate at which this calcification occurs is extraordinary. A healthy coral polyp in ideal conditions can deposit calcium carbonate at a rate that, when multiplied across an entire colony, results in remarkable growth. 1 square meter of healthy reef, covered with vigorously growing coral, processes approximately 35 grams of calcium carbonate daily. That might not sound impressive until you calculate it forward. 35 grams per day becomes 12.8 kilograms per year. Over 10 years, that single square meter of reef produces 128 kilograms of limestone. Over a century, more than 1,000 kilograms. Over a millennium, more than 12 metric tons, from 1 square meter, and the process accelerates dramatically in sunlight. This is where the partnership with zooxanthellae algae reveals its true significance. During the day, when photons rain down through the water, the algae inside coral tissue capture that light energy and convert it into chemical energy through photosynthesis. They produce oxygen as a byproduct, and oxygen saturates the coral tissue, sometimes reaching concentrations 300% higher than the surrounding seawater. They also produce sugars, simple carbohydrates that they share with their coral host. This energy subsidy is substantial. Zooxanthellae can provide up to 90% of the coral's energy needs, freeing the coral from having to catch sufficient prey to fuel its metabolism. But the algae do something else that is perhaps even more important for reef building. Their photosynthesis removes carbon dioxide from the calcification space. Remember that calcium carbonate forms from calcium ions and carbonate ions, but this reaction also produces carbon dioxide as a byproduct. If that carbon dioxide accumulates, it shifts the chemical equilibrium backward, slowing calcification. The zooxanthellae solve this problem by consuming the carbon dioxide as fast as it forms, keeping the reaction running forward at maximum speed. The result is remarkable. Corals with zooxanthellae calcify up to 10 times faster than corals without them. The temperature of the water matters profoundly to all of this chemistry. Coral reefs exist almost exclusively in tropical and subtropical waters, where temperatures remain between 23 and 29 degrees Celsius year-round. This is not coincidence. Below 23 degrees, the enzymes that corals use to concentrate ions and manipulate pH work too slowly. Calcification rates drop. Growth stalls. Above 29 degrees, different problems emerge. The zooxanthellae become stressed, their photosynthesis machinery begins to malfunction, and they start producing reactive oxygen molecules that damage the coral tissue. When this stress continues for too long, corals expel their algae in a desperate attempt to survive, turning white, bleaching, losing their color and their primary energy source simultaneously. The pH of the water is equally critical. Seawater is normally slightly alkaline, with a pH around 8.1 or 8.2. At this pH, carbonate ions are abundant enough for corals to extract what they need. But if the pH drops, if the water becomes more acidic, carbonate ions become scarcer. More of the dissolved carbon exists as bicarbonate or dissolved carbon dioxide instead. Below a pH of 8.0, many corals struggle to calcify at normal rates. Below 7.8, calcification slows dramatically or stops entirely. the limestone skeleton begins to dissolve. This sensitivity to pH has become a source of deep concern in recent decades. The second, oceans are absorbing vast quantities of carbon dioxide from the atmosphere. Carbon dioxide released by human activities. When carbon dioxide dissolves in seawater, it forms carbonic acid, lowering the pH. Since the beginning of the Industrial Revolution, ocean pH has dropped by approximately 0.1 units, from 8.2 to 8.1. That might sound trivial, but pH is measured on a logarithmic scale. A change of 0.1 units represents a 30% increase in acidity, and the change is accelerating. By the end of this century, if carbon emissions continue unabated, ocean pH could drop to 7.8 or below, entering territory where coral calcification becomes severely impaired across all tropical reefs. But return for a moment to that microscopic realm inside the coral polyp, to the cellular mechanisms that make reef building possible. The zooxanthellae themselves are marvels worthy of wonder. Each algal cell measures approximately 10 micrometers in diameter, roughly 1/10 the width of a human hair. Yet within that tiny space exists a complete photosynthetic apparatus, chloroplasts filled with pigments that capture light across the visible spectrum. A single square centimeter of coral tissue, an area no larger than your thumbnail, can contain 1 million zooxanthellae cells. 1 million microscopic solar panels packed into an area smaller than a postage stamp. These algae capture light with astonishing efficiency. In ideal conditions, they can convert up to 95% of photons that strike them into chemical energy. Our best human-engineered solar panels achieve perhaps 25% efficiency. The algae manage nearly 4 times better, and they have been doing so for 50 million years. They accomplish this through an array of pigments that absorb light at different wavelengths, ensuring that virtually no photon escapes unused. Chlorophyll captures red and blue light. Carotenoids capture green and blue. Peridinin, a pigment unique to dinoflagellates, captures blue-green wavelengths that penetrate deepest into water. Together, these pigments harvest the full spectrum of underwater light. The arrangement of these algae within coral tissue is not random. They occupy the upper layers of the polyp, positioned to receive maximum sunlight while still protected by a thin layer of coral tissue above them. The coral must balance competing needs: enough tissue to shield the algae from damaging ultraviolet radiation, but not so much as to block the light they need for photosynthesis. Different coral species achieve this balance in different ways. Some are nearly transparent, allowing maximum light penetration. Others have tissue packed with fluorescent proteins that absorb ultraviolet light and re-emit it as visible light in wavelengths the zooxanthellae can use, effectively converting harmful radiation into useful energy. The chemical communication between coral and algae is constant and complex. The algae produce sugars, primarily glucose, which they actively transport to the coral cells. The coral, in turn, provides the algae with nitrogen and phosphorus, essential nutrients that are scarce in tropical waters. The coral also provides protection from grazers and from the full intensity of sunlight. It is a remarkably balanced exchange, each partner providing what the other needs most. But this partnership has vulnerabilities. The algae are extremely sensitive to temperature, just 2 or 3 degrees above their optimal range, and their photosynthetic machinery begins to fail. The electron transport chains that normally shuttle energy through the chloroplast start producing reactive oxygen species instead, molecules that damage proteins and DNA. When this happens, when the algae become more liability than asset, producing toxins instead of food, the coral makes a desperate choice. It expels them. The mechanism of expulsion is still being studied, but it appears to involve both active rejection by the coral and death of the algae themselves. Within days, sometimes within hours of a heat stress event, corals can expel 90% or more of their zooxanthellae. They turn white, their limestone skeleton visible through the now transparent tissue. Without their algae, corals lose their primary energy source. They must survive on what they can catch, on the tiny zooplankton they snare with their tentacles. Some corals can survive bleached for weeks or even months, waiting for water temperatures to drop, waiting for an opportunity to acquire new algae. But if the stress continues too long, if the heat persists, they starve and die. Yet consider the sophistication of even this catastrophic response. Corals did not evolve the ability to expel zooxanthellae because they faced modern climate change. They evolved it because, over millions of years, they occasionally encountered conditions where keeping the algae was more dangerous than expelling them. The mechanism exists because it sometimes works, because corals that could shed stressed algae and later reacquire healthy ones had a survival advantage. It is a strategy for coping with brief, occasional stress events, not for coping with chronic, sustained warming. The chemistry of calcification also involves trace elements that play crucial roles despite being present in minute quantities. Strontium, for example, substitutes for calcium in the aragonite crystal lattice. The ratio of strontium to calcium in coral skeleton varies with water temperature, a fact that allows scientists to reconstruct past ocean temperatures by analyzing coral cores. Magnesium also incorporates into the skeleton in temperature-dependent ratios. These trace elements do more than just provide a temperature record. They affect the strength and stability of the skeleton itself. Too much magnesium makes aragonite less stable. Too little strontium can weaken the crystal structure. The precision required for all of this to work is extraordinary. The coral must maintain the correct ion concentrations, the correct pH, the correct temperature, all while supporting millions of zooxanthellae, all while catching prey, all while defending against competitors and predators, all while growing and reproducing. It is a performance of staggering complexity, executed by an organism with no brain, no central nervous system, nothing but distributed sensors and ancient genetic programs refined over hundreds of millions of years. And perhaps most remarkably, this entire chemical factory can coordinate with other factories across vast distances. Once each year, in a phenomenon we will explore more deeply later, corals synchronize their reproduction across hundreds or thousands of kilometers. Somehow, they detect environmental cues with extraordinary precision. Water temperature within half a degree, day length within minutes, lunar cycle to the specific night. Then, at exactly the right moment, millions of coral colonies release their gametes simultaneously, turning the ocean into a blizzard of pink and orange spheres. The chemical signals that enable this coordination operate at concentrations measured in parts per trillion. Corals release pheromones, molecular messages that drift through the water, detected by other corals kilometers away. The receptors that sense these molecules are sensitive beyond anything human technology has achieved. We cannot build sensors that detect molecules at parts per trillion in the chaotic environment of the ocean. Yet corals do so reliably, year after year, using those signals to coordinate the timing of their reproduction with precision measured in minutes. All of this extraordinary chemistry, all of this molecular precision, all of this coordinated complexity emerges from a simple polyp, from an organism that consists of just 2 cell layers separated by a thin layer of jelly, from a creature that appears, at first glance, to be among the simplest of animals. But simplicity in form does not imply simplicity in function. The chemical sophistication occurring inside each coral polyp rivals or exceeds anything happening in far more complex organisms. When you swim over a coral reef, when you float above those living cathedrals of limestone, you are witnessing not just biology but chemistry at its finest, molecular machinery operating with a precision that human engineers can barely imagine, let alone replicate. You are seeing the result of 500 million years of refinement, of evolution optimizing every step of a process that turns dissolved minerals into solid stone, that builds structures capable of lasting 10,000 years. that creates habitat for 1/4 of all ocean life. Tomorrow, we will explore those habitats, those living cities built by this extraordinary chemistry. We will travel to the most spectacular coral reefs on Earth, places where the calcification process has been running uninterrupted for thousands of years, where the accumulated limestone reaches depths of hundreds of meters, where the complexity of life defies comprehension. We will follow the explorers who first discovered these underwater wonders, who risked their lives diving into the unknown to reveal the secrets hidden beneath tropical waves. But for now, consider the wonder of what we have just explored. Every coral polyp on every reef in every tropical ocean is, at this very moment, performing this same chemical feat. Millions upon millions of tiny factories, extracting calcium from seawater, concentrating carbonate, depositing aragonite crystal by crystal, building the largest living structures on Earth, atom by atom, ion by ion, day by day, year by year, century by century. It is alchemy of the highest order, the transformation of dissolved minerals into paradise. On a June night in 1770, off the northeastern coast of what would later be called Australia, His Majesty's bark Endeavour struck. Something in the darkness. The collision was not violent, not at first. Captain James Cook, one of history's greatest navigators, had been sailing through uncharted waters, taking soundings regularly, finding reassuringly deep water. Then, without warning, the ship shuddered. It had run aground on something the crew could not see, something lurking just beneath the surface, something that would hold them trapped for nearly a full day. The Endeavour had struck the Great Barrier Reef, a living structure so vast that it stretched for 2,300 kilometers along the Queensland coast, a limestone barrier built by countless trillions of coral polyps over thousands of years. For 23 hours, the ship remained impaled on coral heads, its hull grinding against the limestone as waves rocked it back and forth. The crew threw cannons overboard to lighten the ship. They pumped water frantically as the damaged hull began to leak. Finally, on the next high tide, they managed to float free, but it was a close thing. The voyage of discovery that would map Australia's eastern coast nearly ended on the reef that European science did not yet know existed. Cook's encounter was not the discovery of the Great Barrier Reef. Indigenous Australians had lived alongside it and travelled through it for more than 60,000 years. But it was Europe's violent introduction to the largest living structure on Earth, a structure so massive that it is visible from space, a structure that stretches longer than the Great Wall of China, a structure that is not one reef, but nearly 3,000 individual reefs and 900 islands, woven together in a maze that defies comprehension. To understand the scale of the Great Barrier Reef requires perspective that is difficult to achieve. It covers 344,000 square kilometers of ocean. If you placed it over the United States, it would stretch from New York City to Chicago. If you placed it over Europe, it would cover nearly all of Italy. The limestone beneath the living corals at the surface reaches depths of more than 900 meters in some places, representing thousands of years of accumulated calcification, layer upon layer of coral skeletons building upward toward the light. This vast reef system generates its own weather. The presence of the reef changes how air moves over the region, how moisture accumulates, where clouds form. Studies have shown that the Great Barrier Reef creates clouds that drop 20% more rainfall on coastal areas than would fall without the reef's presence. The reef alters the climate of an entire region, its influence extending far beyond the water. The diversity contained within this structure is staggering. 400 species of coral, 1,500 species of fish, 4,000 species of mollusks, 240 species of birds, 6 species of sea turtle, 30 species of whales and dolphins. But those numbers, as impressive as they are, fail to capture the true complexity. Every species interacts with others, forming networks of dependence and competition, predation and symbiosis. relationships so intricate that scientists are still mapping them centuries after Cook's encounter. 2 centuries after the Endeavour struck the reef, another explorer would revolutionize our understanding of coral reefs, not through collision but through immersion. Jacques Cousteau, the French naval officer who became the world's most famous underwater explorer, descended into the Red Sea in the 1950s with his newly invented aqualung, the device that would make scuba diving possible for millions. What he found beneath the surface defied the expectations of science at the time. The Red Sea reefs grew in conditions that should not have supported them. The water temperatures regularly exceeded 30 degrees Celsius, higher than coral reefs were thought to tolerate. The salinity was extreme, reaching 41 parts per thousand, compared to the ocean average of 35 parts per thousand. Yet the corals thrived, building reefs of extraordinary beauty and complexity. Cousteau discovered coral gardens growing in underwater caves, where sunlight barely penetrated, in blue holes that descended into perpetual twilight, in environments that pushed the boundaries of where reefs were thought to exist. His films brought these underwater worlds to millions who would never dive, revealing for the first time the 3-dimensional complexity of reef structures, the stunning colors of healthy corals, the abundance of life that filled every crevice. He called the ocean the silent world, though in truth it is anything but silent. Reefs produce a constant symphony of sounds, the cracking of pistol shrimp, the grinding of parrotfish beaks, the grunts and croaks of fish communication, the snapping of coral polyps catching prey. But perhaps no dive did more to expand our understanding of reef depth limits than William Beebe's 1934 bathysphere descent off Bermuda. Beebe, a naturalist and explorer, sealed himself inside a steel sphere barely large enough to hold 2 people and was lowered into the ocean on a cable. As he descended past 100 meters, past 200 meters, past 300 meters, he continued to see corals, not the shallow water corals familiar to science, but deep water species adapted to the dim light of the twilight zone. These deep water corals challenged everything scientists thought they knew about reef ecology. Without sufficient light for zooxanthellae to photosynthesize effectively, these corals relied primarily on catching prey, on filtering nutrients from the water. They grew slowly, so slowly that some colonies were later discovered to be thousands of years old. A black coral colony sampled from deep water off Hawaii was radiocarbon dated to 4,265 years old, making it one of the oldest living organisms on Earth, older than the pyramids, older than Stonehenge, older than written language. The exploration of reefs accelerated through the 20th century as diving technology improved. Each advance in equipment allowed scientists to stay deeper and longer, to reach reefs previously beyond human access. The discovery of Indonesia's Raja Ampat archipelago as a biodiversity hotspot came surprisingly late, in the 1990s, when marine surveys revealed that this relatively small region harbored an astonishing concentration of species. Raja Ampat occupies approximately 50,000 square kilometers of ocean in the heart of the Coral Triangle, the region of highest marine biodiversity on Earth. Within this area, scientists have documented 75% of all known coral species, 75%, in just 1% of the Coral Triangle's total area. To put this in perspective, if you wanted to see 3 out of every 4 coral species that exist on Earth, you could see them all by exploring this one archipelago. Over 600 species of hard corals, more than 1,600 species of fish, countless invertebrates, a concentration of biodiversity that staggers the imagination. The reasons for this extreme diversity are complex, involving ocean currents, island geography, geological history, and evolutionary time. The Coral Triangle sits at the confluence of the Pacific and Indian Oceans, where currents from both great ocean basins meet and mix. This region has remained in the warm tropical zone for millions of years, while other reef areas have drifted through climate zones. It has served as a refuge during mass extinctions, a cradle where new species evolve, and a center from which larvae disperse to reefs throughout the Indo-Pacific. Modern exploration has revealed that our knowledge of reefs remains incomplete. Between 60 and 150 meters depth lies a zone called the Mesophotic Reef, the twilight zone, where light is sufficient for some coral growth but too dim for the high-diversity ecosystems we see in shallows. For decades, this zone was largely unexplored, too deep for conventional scuba diving, not interesting enough for deep-sea submersible expeditions. Then, in the 2010s, scientists developed rebreather technology that allowed divers to reach these depths safely for extended periods. What they found astonished them. The Mesophotic zone is not a barren transition area. It harbors unique coral species found nowhere else, fish species new to science, entire ecosystems that had been completely unknown. In Hawaii, scientists diving the Mesophotic zone have been discovering an average of 2 new species per month. 2 species per month, in waters that lie beneath one of the most studied reef systems in the world. The implication is profound. We have barely begun to catalogue the full diversity of reef life. The deepest coral reefs extend down to approximately 150 meters, but coral growth has been documented even deeper. Small colonies and individual polyps have been found at 200 meters, even 300 meters depth in exceptionally clear water. At these extremes, light intensity is less than 1% of surface levels. Photosynthesis is barely possible. The corals that survive here are slow-growing specialists, eking out existence in near darkness, adding perhaps 1 millimeter to their skeleton per year, but return to the shallows, to the sunlit reefs where diversity reaches its peak. The Great Barrier Reef, Raja Ampat, the Red Sea, these famous systems represent only a fraction of Earth's reefs. The Caribbean harbors approximately 65 species of reef-building corals, far fewer than the Indo-Pacific, but including species found nowhere else. Evolutionary experiments isolated when the Isthmus of Panama closed 3 million years ago, separating Atlantic and Pacific populations. The reefs of the Caribbean once rivaled Indo-Pacific reefs in coverage and abundance, though disease, hurricanes, and human impacts have reduced them substantially in recent decades. The Persian Gulf, too hot for most corals, harbors species that have adapted to extreme conditions, corals that routinely experience temperatures exceeding 36 degrees Celsius, temperatures that would kill most reef corals within days. These heat-tolerant populations have become subjects of intense scientific interest as researchers search for genetic traits that might help other corals survive warming oceans. The Hawaiian archipelago, isolated in the middle of the Pacific, 2,000 miles from the nearest continental shore, supports reef ecosystems dominated by endemic species found nowhere else. Approximately 25% of reef fish in Hawaii are endemic, species that evolved in isolation over millions of years. The coral diversity is lower than the Indo-Pacific average, perhaps 60 species, because few larvae can cross the vast oceanic distances to reach these islands. But what Hawaii lacks in species diversity it compensates for with unique adaptations and exceptionally clear water that allows corals to grow in deeper zones than almost anywhere else. Each of these reef systems, each of these underwater cities, has its own character, its own assemblage of species, its own particular beauty, yet they all share fundamental characteristics. They all exist in tropical or subtropical waters. They all depend on the partnership between coral and zooxanthellae. They all build upward toward the light, creating 3-dimensional structures that transform flat seafloors into vertical worlds of extraordinary complexity. The geological history of reefs reveals patterns of growth and drowning, emergence and submergence, driven by sea level changes through ice ages. During the last glacial maximum, 20,000 years ago, when ice sheets locked up vast quantities of water, sea level was 120 meters lower than today. The Great Barrier Reef as we know it did not exist. Corals grew on what is now the continental shelf, far from the current coastline. Then, as the ice melted and sea level rose, the corals grew upward, keeping pace with the rising water, building the structures we see today. Core samples drilled through modern reefs reveal this history layer by layer. At depth, you find coral species adapted to shallow water, species that now live in the sunlit zone. These fossils indicate that the reef has been growing upward for thousands of years, maintaining its position relative to sea level. The rate of upward growth matches the rate of sea level rise, proof of corals' ability to track changing conditions, to build upward fast enough to stay in optimal depth zones. But this ability has limits. If sea level rises too fast, if the rate of rise exceeds the rate at which corals can build upward, the reef drowns, sinking into depths where light is insufficient for growth. This has happened repeatedly through geological time. Drowned reefs discovered by submersibles lie beneath living reefs in many locations. Limestone platforms colonized by deep-water species, monuments to past episodes of rapid sea level rise or coral decline. The exploration of reefs continues today with technology that would seem like science fiction to Cook or Cousteau. Autonomous underwater vehicles map reef structures with sonar, creating three-dimensional models of entire reef systems. Satellites measure water clarity, temperature, and chlorophyll concentrations from space, helping scientists monitor reef health across entire ocean basins. Genetic sequencing allows identification of species from tiny tissue samples, revealing cryptic species that look identical but are genetically distinct, showing us that biodiversity estimates based on visual surveys dramatically undercount true species numbers. Yet for all our technology, for all our scientific instruments and submersibles and satellites, perhaps the most important explorations are still done by divers, by scientists who descend into the water and observe directly, who see the behaviors and interactions that cameras might miss, who develop through experience and intuition for how reef ecosystems function. Every major breakthrough in reef science has come from observation, from someone noticing something unexpected, from the marriage of technology and direct human experience. The reefs we have explored represent the planet's oldest continuously inhabited cities, structures that have been growing for thousands of years, in some cases, tens of thousands in others. The limestone beneath the living veneer of corals at the surface is a record of environmental conditions stretching back through time, each layer preserving information about temperature, sea level, and water chemistry when it formed. Scientists who core through reef structures are like archaeologists, except the history they excavate is not human, but environmental. Not measured in centuries, but in millennia. When you dive a coral reef, when you descend from the surface into that three-dimensional world of color and life, you are entering one of Earth's oldest neighborhoods, a place where organisms have been living, growing, reproducing, competing, cooperating for longer than human civilization has existed. You are seeing the result of calcification processes that have been running uninterrupted for thousands of years, polyp after polyp, adding its contribution, storm after storm, testing the structure, generation after generation, refining the architecture. Tomorrow, we will explore what lives in these limestone cities, the symphony of millions of species that make reefs the most biodiverse ecosystems in the ocean. We will discover organisms so strange they seem invented, relationships so intricate they seem orchestrated, behaviors so specific they seem designed. We will enter the living city and meet its inhabitants, from the smallest microbes to the largest predators, from the loudest shrimp to the quietest cleaners. But tonight, remember this: the reefs that explorers risk their lives to discover, that continue to reveal secrets to scientists who study them, are not merely beautiful. They are ancient, complex, irreplaceable. They are archives of environmental history, libraries of genetic information, laboratories of evolution. They are cathedrals built not by human hands but by the smallest of architects, not in decades but in millennia, not from quarried stone but from minerals extracted atom by atom from seawater. They are wonders worthy of all the risk, all the effort, all the fascination that humans have devoted to exploring them. Coral reefs occupy less than 0.1% of the ocean floor. If you could spread all the coral reefs on Earth across the sea surface, they would cover approximately 284,000 square kilometers, roughly the area of Nevada. Yet within this tiny fraction of ocean live 25% of all marine species, 1/4 of all the fish, all the invertebrates, all the organisms that inhabit the seas make their home on coral reefs. This concentration of diversity is unmatched anywhere in the ocean, unmatched by any ecosystem except perhaps tropical rainforests on land. The reasons for this extraordinary diversity are manifold, but they begin with structure. A coral reef is not a flat surface. It is a three-dimensional labyrinth of caves and crevices, overhangs and passages, holes and hollows at every scale from centimeters to meters. Every square centimeter of reef surface is habitat for something. The top of a coral head, exposed to strong currents and bright light, harbors different species than the underside of that same coral, shaded and protected. The narrow spaces between coral branches provide shelter for small fish. The larger caves beneath massive corals house octopuses, moray eels, and lobsters. This architectural complexity creates countless microhabitats, each supporting its own assemblage of species. But structure alone does not explain the diversity. What makes reefs truly special is the density and intensity of interactions. Every organism on a reef is engaged simultaneously in multiple relationships, eating or being eaten, cooperating or competing, cleaning or being cleaned, protecting or being protected. The network of connections is so intricate that even after a century of scientific study, we are still discovering new relationships, still finding examples of symbiosis and interdependence that nobody suspected existed. Consider the cleaner wrasse, a small fish no longer than your finger. It establishes what scientists call a cleaning station, a specific location on the reef where other fish come to be serviced. The cleaner wrasse picks parasites, dead skin, and bacteria from the bodies of client fish, even entering their mouths and gills to clean these vulnerable areas. A single cleaner wrasse may service 2,000 individual fish daily, removing an estimated 30 million parasites annually from its clients. The relationship is mutualistic. The cleaner gets food, the clients get healthcare. But the sophistication of this interaction extends far beyond simple feeding. Client fish recognize cleaning stations and will travel considerable distances to reach them. They wait in line, sometimes dozens of fish queuing for service. They adopt specific postures that signal their desire to be cleaned, holding their bodies still with fins spread, mouths open, gill plates flared. The cleaners inspect their clients methodically, working over their bodies with movements that appear almost surgical in their precision. And here is the remarkable part. The client fish are often predators, fish that could easily eat the cleaner in a single bite, yet they never do. The cleaning relationship is so valuable that predators suppress their predatory instincts, allowing tiny fish to swim inside their mouths without harm. This is not simple instinct. Studies have shown that cleaner wrasses can recognize individual client fish, remember which clients are regular visitors, and adjust their behavior based on this recognition. They service regular clients more thoroughly than occasional visitors. They prioritize clients that wait patiently over those that appear impatient. They even engage in what appears to be tactical deception, sometimes taking a bite of the healthy mucus layer that protects fish skin rather than removing parasites. When caught cheating, when a client reacts negatively, the cleaner performs what scientists call a reconciliation dance, a specific set of movements that appears to appease the client and restore the relationship. This is cognitive sophistication emerging in an organism with a brain the size of a grain of rice. The cleaning station is just one node in the reef social network. Throughout the three-dimensional city, similar hubs of interaction occur. There are spawning aggregation sites where hundreds or thousands of fish gather annually to reproduce, locations passed down through generations of fish, used for decades or centuries. There are resting areas where nocturnal predators sleep during the day, their presence tolerated by the diurnal fish that would normally be prey. There are feeding zones where specific patterns of current and topography concentrate plankton, attracting swarms of fish that filter the water. And everywhere, constantly, an acoustic symphony plays. Reefs are among the noisiest environments in the ocean. Pistol shrimp, also called snapping shrimp, create the loudest biological sounds in the sea. They possess one enlarged claw with a specialized mechanism that snaps shut at extraordinary speed, producing a cavitation bubble that collapses with a bang, measuring 210 decibels, louder than a gunshot, louder than a jet engine at close range. For the microsecond during which that bubble collapses, the temperature inside reaches approximately 4,000 degrees Celsius, briefly matching the surface temperature of the sun. The shrimp use this sonic weapon to stun prey and defend territory. When thousands of pistol shrimp inhabit a reef, their collective snapping creates a crackling chorus audible from boats on the surface. Parrotfish add percussion to the symphony. These colorful fish, named for their beak-like teeth fused into a solid plate, bite directly into coral skeletons, grinding the limestone to extract the algae living within. The sound of parrotfish feeding is distinctive, a crunching that carries through the water. Each bite removes a chunk of coral skeleton. The fish digest the algae and excrete the limestone as fine sand. A single large parrotfish can process 5 tons of coral skeleton annually, producing most of the white sand that forms tropical beaches. The beach you walk on in the Maldives or the Caribbean is largely parrotfish excrement, the ground-up skeletons of corals processed through the guts of these remarkable herbivores. The grinding of parrotfish beaks is audible from 30 meters away. But they are not the only source of reef music. Fish produce sounds for communication using specialized muscles that vibrate their swim bladders, creating grunts, croaks, chirps, and pops. Different species have different vocalizations, and some fish can recognize the calls of their own species, using sound to locate mates or maintain school cohesion in murky water or at night. The combined effect of pistol shrimp snapping, parrotfish crunching, fish vocalizing, waves breaking on the reef crest, and countless other sounds creates an acoustic landscape so complex that marine biologists can identify healthy reefs by their soundscapes alone. This noise is not merely interesting, it is functional. Larval fish drifting in open ocean use reef sounds to navigate towards suitable settlement habitat. Experiments have shown that larvae swim preferentially toward recordings of healthy reef soundscapes. The symphony of the reef is a beacon, guiding the next generation home. At night, the reef transforms completely. As the sun sets, approximately 70% of the fish that were active during the day retreat into crevices, wedging themselves into spaces so tight that predators cannot extract them. Many produce mucus cocoons, envelopes of secreted slime that may help mask their scent from nocturnal hunters. As diurnal fish disappear, nocturnal species emerge. Moray eels slide out from their daytime hiding spots. Octopuses hunt across the reef, their flexible bodies squeezing through impossibly small openings. Soldierfish and squirrelfish, with their enormous eyes adapted to collect maximum light, to patrol the darkness. The eyes of nocturnal reef fish are remarkable organs. They contain densities of rod cells, the photoreceptors responsible for light detection, that are among the highest in the animal kingdom. These cells can capture 99% of photons that strike them, allowing the fish to see in light levels that appear pitch black to humans. Some species also possess a reflective layer behind the retina, called a tapetum lucidum, the same adaptation that makes cat eyes shine in flashlight beams. Photons that pass through the retina once reflect back for a second chance at detection, doubling the effective sensitivity. But perhaps the most extraordinary visual system on any reef belongs to the mantis shrimp. These ambush predators, which despite their name are not true shrimp but stomatopods, possess eyes unlike any others on Earth. Each eye contains 16 types of color receptors compared to the 3 types humans possess. This does not mean mantis shrimp see 16 primary colors. Rather, they possess a color recognition system so different from ours that we struggle to comprehend it. They can detect polarized light, seeing patterns that are completely invisible to us. They can detect ultraviolet and infrared wavelengths. Each eye can move independently and can perceive depth on its own, meaning each eye effectively has depth perception without needing the other eye for triangulation. These remarkable eyes serve remarkable predators. Mantis shrimp come in 2 basic types, spearers and smashers. Spearers have raptorial appendages lined with spikes that they thrust into soft-bodied prey with incredible speed. Smashers have club-like appendages that they use to pound hard-shelled prey, striking with accelerations exceeding 10,000 times gravity. The strike is so fast that it creates cavitation bubbles similar to those produced by pistol shrimp. When a smasher-type mantis shrimp punches a snail or crab, the impact can shatter aquarium glass, fracture human fingers, or split the prey's shell completely. The force delivered is out of all proportion to the animal's size. A peacock mantis shrimp measuring 15 centimeters can strike with a force exceeding 1,500 newtons, comparable to a .22 caliber bullet. The diversity of reef inhabitants includes organisms at every size scale, from bacteria visible only under microscopes to whale sharks measuring 12 meters or more. The microbial communities living within coral tissue and skeleton represent their own universe of diversity, with recent genetic sequencing revealing thousands of bacterial species in a single coral colony. These microbes are not passive riders. They cycle nutrients, produce vitamins, defend against pathogens, and likely perform countless other functions we are only beginning to understand. The coral microbiome, as it is called, may be as important to coral health as the zooxanthellae algae, another layer of symbiosis and interdependence. The chemical warfare conducted on reefs is largely invisible but intense. Corals compete for space using specialized sweeper tentacles that can extend far beyond their normal reach. These tentacles, loaded with nematocysts, the stinging cells that are the signature weapon of cnidarians, inject toxins into competing corals that contact them. The toxins kill tissue, clearing a zone around the aggressive coral. Different coral species have different levels of aggression. Some are peaceful, some highly aggressive. When 2 corals of different species grow toward each other, the outcome depends on their relative aggression and their ability to deploy chemical and physical weapons. But corals are not the only organisms engaged in chemical warfare. Sponges produce some of the most toxic compounds in the ocean, molecules that deter predators and inhibit the growth of competing organisms. Nudibranchs, colorful sea slugs that feed on sponges and corals, sequester these toxins in their own tissues, making themselves poisonous to predators. Some nudibranchs even steal the nematocysts from corals they eat, incorporating these stinging cells into their own skin as a defense mechanism. The nudibranch does not digest the nematocyst but transports it intact to specialized storage organs in its skin, where the cell remains functional, ready to fire at anything that attacks the slug. The feeding strategies on reefs span every possibility. Some fish are strict herbivores, grazing algae from rock surfaces. Others are planktivores, picking tiny animals from the water column. Still others are corallivores, feeding exclusively on coral polyps, using specialized mouths and digestive systems to process coral tissue while excreting the limestone skeleton. Butterfly fish and some angelfish specialize in coral feeding, their elongated snouts allowing them to reach into coral branches to nip individual polyps. Some species, like the crown of thorns starfish, are devastating coral predators, capable of eating vast areas of reef. A single large crown of thorns can consume 6 square meters of living coral per year. When populations explode into outbreaks, thousands of these starfish can devastate entire reef sections, leaving only white limestone skeletons behind. Yet even these destructive outbreaks play a role in reef ecology. They create disturbance that opens space for new coral recruitment, potentially increasing diversity by preventing any single coral species from dominating completely. The debate over whether crown of thorns outbreaks are natural or human-caused continues, with evidence suggesting that both natural cycles and human impacts like nutrient pollution may play roles. The symbiotic relationships on reefs extend far beyond coral and zooxanthellae. Anemones house clownfish that defend their hosts from predators and provide nutrients through their waste products. Gobies live in burrows dug by nearly blind pistol shrimp, acting as lookouts while sharing the shrimp's home. Large fish allow smaller fish to remove parasites from their skin. Hermit crabs carry anemones on their shells, gaining protection from the anemone's stinging tentacles while providing the anemone with mobility and access to food scraps. The examples multiply endlessly, each species embedded in a web of relationships that define its existence. The temporal patterns of reef life add another dimension to the complexity. Daily cycles of day and night drive major shifts in species activity. Lunar cycles trigger reproductive events, with many species timing their spawning to specific moon phases. Annual cycles bring seasonal visitors, manta rays arriving to feed on plankton blooms, whale sharks migrating through to filter spawn, turtles returning to nest on beaches adjacent to reefs. Some fish, like the groupers that form spawning aggregations, return to the same location on the same date year after year, guided by internal calendars of extraordinary precision. The age structure of reef communities reveals histories spanning decades or centuries. The largest coral colonies may be hundreds of years old, their size a testament to centuries of successful growth. These massive corals serve as habitat for countless smaller organisms, their crevices and overhangs providing shelter for generations of fish and invertebrates. When one of these ancient corals dies, whether from bleaching, disease, or physical damage, an entire community loses its foundation. The ecological consequences ripple outward, affecting species that depended on that specific coral, and species that depended on those species, cascading through the food web. The productivity of reefs, their ability to generate biomass from sunlight and nutrients, rivals any ecosystem on Earth. In the nutrient-poor waters of tropical oceans, reefs are oases of abundance, their productivity seeming to violate the rules of ecology. The secret lies in the efficiency with which reefs retain and recycle nutrients. Very little is wasted. When a fish defecates, its waste is captured by filter feeders before it can leave the reef. When organic material dies, it is decomposed by bacteria and consumed by detritivores. Its nutrients return to the system. The tight coupling between organisms keeps nutrients cycling within the reef rather than being lost to the surrounding ocean. This productivity supports food webs of extraordinary complexity. Primary producers, mainly the zooxanthellae in corals plus various algae, form the base. Herbivores feed on algae. Small carnivores feed on herbivores. Larger carnivores feed on smaller carnivores. But the reality is far more complex than this simple chain suggests. Most reef organisms eat multiple food types. Most participate in different feeding roles at different life stages. The juvenile parrotfish feeds on small invertebrates. The adult grinds coral. The Nassau grouper starts life feeding on plankton, transitions to small fish as a juvenile, and becomes an apex predator as an adult. The network of who eats whom is not a chain or even a web, but a tangled, multidimensional matrix of interactions. At the top of this matrix sit the apex predators, large groupers, sharks, barracudas. These predators regulate prey populations, preventing any single species from becoming so abundant that it destabilizes the system. Reefs without apex predators look different from reefs with them. The prey fish behave differently, venture into different areas, feed at different times. The removal of apex predators cascades through the entire ecosystem, a demonstration of how interconnected reef communities truly are. Yet the most numerous predators on the reef are not the impressive sharks or groupers, but the tiny planktivorous fish that school in huge aggregations above the reef, feeding on the microscopic animals drifting in the current. Chromis, Anthias, and Fusiliers, by the thousands, create living clouds above coral formations, their collective feeding, the processing enormous quantities of plankton. These small fish, in turn, become prey for larger fish, transferring energy from the open water to the reef community. The reef is, fundamentally, a city. It has neighborhoods where different communities live. It has traffic patterns as organisms move between feeding and resting areas. It has a 24-hour rhythm of activity. It has infrastructure in the limestone framework that everyone depends on. It has service industries in the cleaning stations and the detritivores that handle waste. It has construction workers in the corals that maintain and expand the infrastructure. It has security in the predators that maintain order. It has invisible utilities in the bacteria and other microbes that cycle nutrients and process waste. This city operates without any central planning, without any government, without any architect designing the system. It is the product of evolution, of countless species, each pursuing its own survival and reproduction, each inadvertently contributing to the functioning of the whole. The complexity emerges from simple rules repeated billions of times. Eat without being eaten, reproduce more successfully than competitors, adapt to changing conditions. From these simple rules, executed by organisms ranging from bacteria to sharks, emerges the most biodiverse ecosystem in the ocean, a symphony of 10 million species playing together, each contributing its note to a composition 400 million years in the making. Tomorrow, we will confront the reality that this symphony is under threat, that the reef city faces challenges unprecedented in the long history of coral evolution. We will explore the bleaching events that have devastated reefs worldwide, but also the remarkable discoveries of resilience, the corals that survive where others die, the scientists working to preserve these ecosystems, and the reasons we must not give up hope. But tonight, if you close your eyes and imagine a coral reef, do not picture a static backdrop, a colorful rock formation. Picture a living city in constant motion, where every centimeter is inhabited, where every moment something is being born or dying, eating or being eaten, where thousands of species are simultaneously cooperating and competing, where the density of life and interaction exceeds anywhere else in the sea. Picture a place so complex that science has studied it for centuries, yet still discovers new species monthly, still finds relationships nobody suspected, still uncovers mechanisms nobody understood. Picture paradise, built by the smallest architects, inhabited by millions, playing a symphony that has echoed through the oceans for longer than forests have grown on land. The ocean temperature has risen 0.8 degrees Celsius since 1900, less than 1 degree. In our daily experience, such a small temperature change would be imperceptible. The difference between a room at 23 degrees and 23.8 degrees, utterly unnoticeable to human sensation. But for coral reefs, which evolved over millions of years in waters that fluctuated within a narrow range, this seemingly trivial warming represents a catastrophic shift. Bleaching events, once so rare they might occur once per century at any given location, now strike the same reefs every 6 years on average. The mathematics of this acceleration should alarm us. An event that was extraordinary, that most individual coral colonies would never experience in their lifetimes, has become routine, expected, inevitable. Corals that evolved to handle occasional stress now face repeated stress, chronic stress, relentless pressure that allows no time for recovery between events. The 1998 El Niño brought water temperatures far above normal across the tropical Pacific and Indian Oceans. The heat persisted for months, from December through May, subjecting corals to sustained thermal stress beyond anything in recent history. When it ended, 16% of the world's coral reefs were dead. 16%. In 9 months, entire reef systems in the Maldives, the Seychelles, throughout the Indian Ocean suffered mortality exceeding 90%. Reefs that had been building for thousands of years were reduced to rubble, their living veneer stripped away, their limestone skeletons exposed to the elements. The mechanism of bleaching is now well understood, though this understanding does nothing to make it less tragic. When water temperature exceeds the tolerance range of zooxanthellae, typically around 30 to 31 degrees Celsius for most corals, the photosynthetic machinery inside the algae begins to malfunction. Instead of producing energy and oxygen, the algae start generating reactive oxygen species, molecules that are toxic to both the algae and the coral. The coral faces a choice: keep the algae and suffer increasing tissue damage from the toxins they produce, or expel the algae and lose the energy source that provides up to 90% of its nutrition. Evolution has provided corals with the machinery to expel zooxanthellae, because there have always been occasional situations where this desperate measure was necessary. But the mechanism evolved to handle rare, brief stress events. It did not evolve to handle sustained warming, year after year, with insufficient recovery time between bleaching events. A coral can survive bleached for weeks, perhaps months if it is healthy and conditions are good, but repeated bleaching exhausts the coral's reserves, weakens its immune system, reduces its ability to compete with algae that grow over stressed colonies. The third bleaching is often fatal, even if the first 2 were survived. 18 years after the 1998 event, an even more severe bleaching struck. In 2016, temperatures across the Great Barrier Reef exceeded averages by 2 degrees Celsius for months. The northern section of the reef, the most pristine portion, suffered worst. When scientists surveyed after the event, they found 50% mortality in the northern third of the reef. 50%. Half of the corals in the best preserved section of Earth's largest reef system died in 2 months. The surveys revealed scenes of devastation, vast fields of white coral skeletons, the algae gone, the tissue dying or dead, the framework remaining but the life extinguished. In 2017, before the reef could recover, before survivors could reproduce and replenish dead areas, another bleaching event struck. Then again, in 2020, 3 major bleaching events in 5 years on a reef that previously might have experienced bleaching once per century. The cumulative impact is catastrophic. Coral cover on the Great Barrier Reef has declined by approximately 50% since 1985, with the majority of that loss occurring after 2000. Yet even in this litany of devastation, hope emerges from unexpected places. Scientists studying reefs in the Persian Gulf discovered coral populations thriving in conditions that should be lethal. Water temperatures in the southern Persian Gulf regularly reach 36 degrees Celsius in summer, 8 to 10 degrees above the temperature that kills most reef corals. Yet the corals here have adapted. They possess zooxanthellae strains that can photosynthesize effectively at extreme temperatures. Their cellular machinery is calibrated differently, able to maintain function where others fail. These heat-tolerant corals did not evolve in the past century. They are the product of thousands or tens of thousands of years of natural selection in an extreme environment. But their existence proves that corals can adapt to higher temperatures, given sufficient time. The question is whether adaptation can occur fast enough to match the pace of current warming. In nature, adaptation to new conditions typically requires hundreds or thousands of generations. Corals can live for decades or centuries, so their generation time is long. The ocean is warming faster than natural adaptation can likely occur for most species. This realization has driven scientists to explore interventions, ways to help corals adapt faster than they could naturally. The Coral Restoration Foundation in Florida operates underwater nurseries where they grow staghorn and elkhorn corals, species that have declined by more than 97% in the Caribbean over the past 40 years. The nurseries consist of trees made of PVC pipe suspended in the water, with coral fragments attached to grow. Each fragment, carefully tended by divers, grows into a colony over 1 to 2 years. The foundation then transplants these laboratory-raised corals onto degraded reefs. They now grow approximately 40,000 corals annually, outplanting thousands of colonies onto reefs throughout the Florida Keys and Caribbean. The survival rate of outplanted corals is improving as techniques are refined, now exceeding 90% in many locations. But the scale remains challenging. Growing 40,000 corals per year is impressive for a single organization, but the Caribbean contains millions of square kilometers of reef. Even successful restoration at current scales would take centuries to restore what has been lost. The work is essential, providing local benefits and refining techniques, but it cannot by itself solve the crisis. Genetic rescue offers another approach. Researchers in Hawaii are selectively breeding corals, crossing individuals from different reefs to create offspring that might be more resilient to warming and acidification. They subject young corals to elevated temperatures, identifying which individuals survive best, then using those survivors as breeding stock for the next generation. This accelerated selection mimics natural adaptation, but compresses thousands of years of evolution into decades. Early results are promising. Corals bred for heat tolerance show significantly higher survival rates when exposed to temperatures that kill wild corals. Similar work is exploring acid tolerance. As oceans absorb carbon dioxide from the atmosphere, the water becomes more acidic, making carbonate ions less available for calcification. By the end of this century, if emissions continue unabated, ocean pH could drop to levels that severely impair coral calcification worldwide. Researchers are identifying corals that maintain calcification rates in acidified water and breeding from these tolerant individuals. Some corals from naturally acidic environments, like volcanic vents where carbon dioxide seeps from the seafloor, show remarkable tolerance to conditions projected for 2100. The genetic tools available to researchers have advanced dramatically. It is now possible to sequence a complete coral genome in days rather than years, to identify specific genes associated with heat tolerance or disease resistance, to compare genomes across populations and species. This genetic information is revealing that coral resilience often correlates with genetic diversity. Populations with high genetic diversity tend to weather bleaching events better than genetically uniform populations, because in diverse populations, some individuals possess alleles that confer heat tolerance. This finding has profound implications for restoration. Rather than cloning a few successful individuals, restoration efforts should maintain and enhance genetic diversity, outplanting corals from multiple sources, ensuring that restored populations contain the genetic variation necessary to adapt to changing conditions. It is a more complex approach than simply growing more corals, but it is more likely to create resilient populations capable of surviving future challenges. Traditional ecological knowledge is informing modern conservation in surprising ways. In Fiji, indigenous communities have practiced taboo for centuries, temporary fishing closures on specific reef areas to allow populations to recover. When these traditional practices are implemented with modern scientific monitoring, the results are remarkable. Fish populations in taboo areas can recover 500% within 3 years, their increased abundance spilling over to benefit adjacent fished areas. The reef ecosystems become more resilient, better able to weather bleaching events and other stresses when their fish populations are healthy and diverse. The connection between fish populations and coral health is direct. Herbivorous fish control algae that compete with corals for space. When overfishing removes these herbivores, algae can overgrow stressed or dead corals, preventing recovery. Reefs with healthy herbivore populations recover faster from bleaching than reefs where herbivores are depleted. This is practical knowledge that indigenous communities have understood for generations. Protecting fish protects reefs, and protecting reefs protects fish. The cycle is reciprocal. Engineering solutions are being explored. Alongside biological approaches, scientists are experimenting with artificial structures designed to accelerate coral settlement and growth. Three-dimensional printed reef structures made from calcium carbonate or similar materials provide substrate that corals colonize 50 times faster than natural recruitment onto dead reef. The printed structures can be designed with optimal complexity, creating the caves and overhangs that fish and invertebrates require, while maximizing the surface area available for coral settlement. Researchers are also investigating cloud brightening, a technique that could potentially cool reef waters during heat waves. The concept involves spraying microscopic seawater droplets into the air above reefs, creating clouds of tiny particles that reflect sunlight, reducing the amount of solar energy reaching the water. Early experiments over the Great Barrier Reef suggest this could lower water temperatures by up to 2 degrees Celsius in treated areas. The technology is still experimental, and questions remain about effectiveness, cost, and potential unintended consequences. But if it works, it could provide a tool to protect critical reefs during extreme heat events, while longer-term solutions are implemented. Probiotics for corals represent another frontier. Just as humans consume beneficial bacteria to improve gut health, corals might benefit from supplementation with beneficial microbes. Researchers are identifying bacterial strains that improve coral disease resistance or help corals tolerate heat stress. These bacteria could potentially be cultured and sprayed over reefs, inoculating corals with microbial partners that enhance their resilience. The approach is still in early research stages, but initial results suggest some bacterial strains do improve coral survival under stress. The most controversial proposed intervention is genetic modification. Could scientists edit coral genomes to insert heat tolerance genes, disease resistance genes, or other traits that would improve survival? The technology to do this exists. The ethical questions are profound. Should we modify organisms to adapt them to human-caused changes, rather than addressing the root causes of those changes? Would genetically modified corals successfully integrate into reef ecosystems, or might they have unintended effects? Would they be accepted by the public and policymakers? These questions have no easy answers, but they are being asked because the alternatives are grim. If current trends continue, if oceans keep warming at present rates, most coral reefs could effectively cease to exist as functional ecosystems by mid-century. Not completely extinguished, but degraded to the point where they no longer support diverse communities, no longer provide coastal protection, no longer function as nurseries for fish, no longer generate the resources that hundreds of millions of people depend upon. The economic value of reefs is calculated at $375 billion annually through tourism and fishing alone. 500 million people depend directly on reefs for food and livelihoods. The stakes of reef conservation extend far beyond ecology into economics, food security, and human well-being. Yet the ultimate solution to reef decline is not technological but political. All the restoration projects, all the genetic rescues, all the engineering interventions are buying time, treating symptoms rather than causes. The fundamental driver of reef decline is greenhouse gas emissions warming the planet. Until emissions decline substantially, until atmospheric carbon dioxide concentrations stabilize and ocean temperatures stop rising, coral reefs will continue to decline. The interventions being developed are essential, keeping corals alive through the warming, maintaining genetic diversity and reef function, but they cannot substitute for addressing the root cause. There is precedent for successful conservation, changing trajectories that seemed irreversible. In 1985, scientists discovered a hole in Earth's ozone layer above Antarctica, caused by chlorofluorocarbons used in refrigerators and aerosols. The ozone layer protects life from ultraviolet radiation, and its loss threatened catastrophic increases in cancer rates and ecosystem damage. The world responded with the Montreal Protocol, an international agreement that phased out ozone-depleting chemicals. Today, the ozone hole is shrinking. The protocol is considered the most successful environmental treaty ever negotiated. It demonstrates that humans can coordinate globally to address planetary-scale environmental threats when the danger is clear and solutions are available. The challenge with coral reefs is that the solutions require transforming the global energy system, moving from fossil fuels to renewable sources, a task far more complex than replacing refrigerant chemicals. But the alternative, the loss of coral reefs and the consequences that cascade from that loss, is unacceptable. Reefs are not expendable. They are irreplaceable repositories of biodiversity, magnificent examples of evolutionary creativity, essential ecosystems supporting millions of species and billions of human livelihoods. The corals surviving in the Persian Gulf at 36 degrees, the populations recovering after bleaching events, the restoration projects succeeding in replanting reefs, the communities protecting their reefs through traditional practices, all of these demonstrate that the situation is not hopeless. Corals have proven over 500 million years that they can survive catastrophic change. They have weathered mass extinctions, ocean acidification events, asteroid impacts. They can survive the challenges of the 21st century, but only if we give them the chance. Only if we act quickly enough to limit warming. Only if we protect them from the additional stresses of overfishing, pollution, and physical destruction while they adapt to changing conditions. Tomorrow, we will witness one of nature's most spectacular events, the mass spawning of corals on the Great Barrier Reef, a synchronized reproductive phenomenon that shows how these ancient organisms still possess capabilities that astonish science. But tonight, understand that the race against time is not yet lost. The window for action remains open. The tools for conservation are being developed and deployed. The knowledge of what must be done is clear. What remains uncertain is whether humanity will act with sufficient speed and scale to preserve these underwater cathedrals, these living monuments to evolution's creativity, these paradise gardens that paint the tropical seas with color and fill them with life beyond measure. Once each year, triggered by the full moons of October and November, the Great Barrier Reef stages the largest synchronized reproductive event on Earth. For a few nights during these specific moon phases, when conditions align precisely, billions of coral colonies release their gametes simultaneously, transforming the ocean into an underwater snowstorm flowing upward, a blizzard of pink and orange spheres rising through the dark water toward the surface. It is one of nature's most spectacular phenomena, a mass spawning event so precisely coordinated across such vast distances that scientists are still working to understand how corals achieve it. The timing is exquisitely specific. Spawning occurs exactly 120 minutes after sunset. Not approximately 2 hours, not roughly after dark, but 120 minutes, give or take just a few minutes. This precision holds across hundreds of kilometers of reef, with colonies separated by vast distances releasing their gametes within minutes of each other. The cues that trigger spawning are multiple and interlocking. Water temperature must be stable at approximately 27 degrees Celsius for at least a week. Day length must match October or November photoperiods. The moon must be full or nearly full, and even with all these conditions met, not all corals spawn every month. Some wait for October, others for November. Some spawn on the full moon, others 4 to 6 days after. The patterns are species-specific, consistent across years, yet flexible enough that corals can adjust timing if conditions are not ideal. The mechanism by which corals sense these environmental cues is remarkable. They possess light-sensing proteins called cryptochromes, the same molecules that birds use for magnetic navigation during migration. In corals, cryptochromes detect both sunlight and moonlight, allowing the coral to track lunar cycles even underwater, even at depth where moonlight is barely perceptible to humans. The corals are essentially keeping a calendar, counting the days between moon phases, measuring photoperiod to determine what month it is, monitoring temperature to assess whether conditions are suitable. But how does this information translate into synchronized spawning? How do colonies separated by kilometers coordinate their reproduction to within minutes? The answer lies in chemical communication, operating at concentrations almost beyond measurement. Corals release pheromones, molecular signals that drift through the water, detected by other corals kilometers away at concentrations measured in parts per trillion. When the first corals begin to spawn, their pheromones signal others that the time has come. The spawning cascades across the reef like a chain reaction, each colony's release triggering its neighbors, the wave of reproduction spreading outward until thousands of square kilometers of reef are spawning simultaneously. The preparation for spawning begins months in advance. Inside each polyp, in the tissue layers between the stomach cavity and the skeleton, packets of eggs and sperm develop slowly. The eggs are large by cellular standards, hundreds of micrometers in diameter, packed with yolk to nourish the developing embryo after fertilization. The sperm are tiny, typical for animals, consisting of little more than genetic material and a tail for swimming. Corals produce both simultaneously, hermaphroditic organisms that generate both egg and sperm within the same colony, though the mechanisms that prevent self-fertilization ensure that most eggs are fertilized by sperm from other colonies. As spawning night approaches, these gamete bundles, each containing both eggs and sperm wrapped in a membrane, move toward the polyp's mouth. They pack into the gastric cavity, sometimes filling it so completely that the polyp appears swollen. Then, at the appointed time, 120 minutes after sunset, the mouth opens and the bundles float upward. The release is not violent or sudden. The bundles emerge gently, rising through the water column at approximately 3 centimeters per second, their buoyancy carefully calibrated to ensure they reach the surface without dispersing too widely. A single large brain coral, a colony perhaps 2 meters across, can release 4 million eggs in 30 minutes. 4 million from one colony. Multiply this by the millions of colonies across the Great Barrier Reef, and the numbers become incomprehensible. Trillions of gamete bundles, each containing dozens of eggs and millions of sperm, rising through the water simultaneously, creating a reproductive event visible from research vessels on the surface, where observers describe the water as looking like a blizzard, like snow falling upward through the beams of dive lights. The surface of the ocean during spawning becomes covered with a slick of spawn, pink and orange and red, depending on the coral species, the colors coming from carotenoid pigments in the eggs. This slick can be meters thick, creating rafts of reproductive material that drift with surface currents. Within these rafts, the choreography of fertilization unfolds. The gamete bundles break apart at the surface, releasing eggs and sperm into close proximity. The sperm swim toward eggs, guided by chemical attractants that eggs release, following concentration gradients to their targets. Each egg might be approached by thousands of sperm, but only one will successfully fertilize it. The timing of spawning maximizes the probability of successful fertilization while minimizing the probability of predation. By spawning synchronously, corals create a situation where there is far too much spawn for predators to consume it all. This is predator satiation, overwhelming the capacity of filter feeders and planktivores to eat the available food. No matter how many gametes are consumed, millions more escape. The strategy works. Despite the feast that spawning provides to reef inhabitants and visiting predators, the sheer volume of spawn ensures high fertilization rates and widespread larval dispersal. Predators do gather for the event. Whale sharks, the largest fish in the ocean, arrive at the Great Barrier Reef precisely on schedule, their 8-meter bodies perfectly adapted to filter feeding. They swim through the spawn slicks with mouths open, filtering tons of protein-rich gametes through their gill rakers. These gentle giants travel thousands of kilometers to be present for spawning, suggesting they either remember the timing from previous years or can detect chemical cues in the water that signal the approaching event. Other planktivores converge as well: manta rays, schools of fusiliers, countless smaller fish, all gorging on the abundance. Yet for every gamete consumed, thousands escape. Within hours of release, fertilization rates often exceed 70%, a remarkable success given the dilution of sperm in open water and the complexity of bringing egg and sperm together in a 3-dimensional ocean environment. The fertilized eggs begin dividing immediately, each cell division bringing them closer to becoming a planula. Larva, the free-swimming stage that will eventually settle onto the reef and begin building a new coral colony. The larvae that develop from successfully fertilized eggs possess capabilities that seem impossible for such simple organisms. Each planula is approximately 0.5 millimeter long, elongated and covered with tiny hairs called cilia that beat in coordinated waves to propel it through the water. Inside this microscopic package is everything needed to sense the environment, navigate through the ocean, find suitable settlement habitat, and transform into a coral polyp capable of building a limestone skeleton. The larva contains no brain, no eyes, no complex sensory organs, yet it can detect light levels, chemical signatures, water flow patterns, and surface textures. For days or weeks, depending on the species, these larvae drift in the ocean currents. They are at the mercy of the water's movement, yet they are not entirely passive. They can swim vertically, rising toward the surface or sinking toward the bottom, positioning themselves in different current layers to influence where they are transported. This behavior, called ontogenetic vertical migration, allows larvae to exert some control over their dispersal, though the dominant force remains the currents themselves. The dispersal of coral larvae connects reef systems across vast distances. Larvae spawned on one reef can settle on another reef hundreds of kilometers away, creating genetic links between populations that would otherwise be isolated. This connectivity is essential for reef resilience. When a reef is damaged by bleaching, storm, or disease, its recovery often depends on larvae arriving from distant healthy reefs to recolonize dead areas. The network of reefs throughout a region functions not as isolated ecosystems, but as a metapopulation, a connected system where the fate of each reef influences and is influenced by the fates of others. Ocean currents act as highways for larval dispersal, transporting spawn from upstream reefs to downstream locations. The direction and strength of these currents determine which reefs are sources, producing more larvae than they receive, and which are sinks, receiving more larvae than they produce. Understanding these patterns is critical for conservation. Protecting source reefs, the upstream locations that seed downstream reefs with larvae, is more important than protecting sinks. Yet identifying sources and sinks requires detailed knowledge of currents, spawning times, larval behavior, and settlement patterns, knowledge that scientists are still assembling. When a larva finds suitable habitat, when it encounters a surface that provides the chemical and physical cues indicating a good settlement location, it undergoes metamorphosis. The free-swimming larva transforms into a sessile polyp, attaching permanently to the substrate. This transformation is irreversible and immediate. The larva cements itself to the rock using adhesive proteins, loses its cilia, develops tentacles and a mouth, and begins secreting the skeleton that will be its foundation and fortress for the rest of its life. The choice of settlement location is perhaps the most important decision a coral makes. The polyp will spend its entire life in that exact spot, unable to move if conditions deteriorate, unable to escape predators or competition. Evolution has equipped larvae with sophisticated settlement preferences. They prefer surfaces already colonized by specific types of coralline algae, red algae, that produce chemical cues indicating favorable conditions. They avoid surfaces covered with fleshy algae or sediment. They settle preferentially in crevices and on vertical surfaces where they are protected from predation and sedimentation. They appear to avoid areas where other corals are already abundant, perhaps detecting chemical signals indicating intense competition. Once settled, the polyp begins the work of calcification, depositing aragonite beneath its tissue, building the limestone cup that will protect and support it. Within days, it may undergo its first division, budding into 2 polyps, then 4, then 8. The colony grows through repeated asexual reproduction, each polyp a clone of the original settler. Growth rates vary enormously depending on species, environmental conditions, and genetics. Fast-growing branching corals like staghorn can add 15 centimeters or more per year under ideal conditions. Massive, slow-growing corals like brain corals might add only 5 millimeters per year. But the massive corals can live for centuries, eventually reaching sizes exceeding several meters in diameter, while the fast-growing branching corals might live only decades before a storm breaks them apart. The synchronization of spawning extends beyond just timing to the number and ratio of gametes produced. Corals adjust the number of eggs they produce based on their size and condition. Larger colonies produce more eggs. Healthier colonies produce larger eggs with more yolk reserves. The ratio of eggs to sperm within each bundle is also regulated, typically with 1 egg surrounded by hundreds of thousands of sperm. This ratio ensures that when bundles break apart at the surface, sperm are abundant enough to find and fertilize eggs, but not so abundant that sperm competition wastes resources. The energy investment in reproduction is enormous. For coral colonies preparing to spawn, up to 50% of their annual energy budget may be devoted to gamete production. This massive investment means that spawning is costly, a risk that only pays off if conditions are right for successful fertilization and larval survival. This is why the environmental cues must align precisely. Spawning in the wrong conditions, when temperatures are too high or too low, when currents are unfavorable, when predator abundance is exceptionally high, would waste the enormous energy investment with little chance of reproductive success. Scientists studying coral spawning have discovered additional layers of complexity. Some corals spawn multiple times per year, not just during the mass spawning event. These opportunistic spawners release smaller batches of gametes at different times, perhaps hedging their bets, ensuring that at least some reproductive events might succeed even if the mass spawning fails. Other corals are brooders rather than broadcasters. Instead of releasing eggs into the water, they retain eggs internally, release only sperm, and internal fertilization produces larvae that the coral releases already developed, ready to settle within hours or days. Brooding species sacrifice dispersal distance for increased larval survival, a different reproductive strategy that works well in some environments. The genetics of spawned larvae reveal patterns of relatedness that inform our understanding of reef connectivity. By sampling larvae and comparing their DNA to potential parent colonies across multiple reefs, scientists can track which larvae came from which reefs, mapping the pathways of larval dispersal. These genetic studies have revealed that most larvae settle relatively close to their parent reef, within tens of kilometers, but a small percentage travel much farther, sometimes hundreds of kilometers, providing the occasional long-distance dispersal that maintains genetic connectivity across entire regions. The mass spawning phenomenon was only discovered by science in 1981 on the Great Barrier Reef, by researchers who were diving at night and witnessed it by chance. Before that night, scientists knew that corals reproduced, but assumed the process was asynchronous, with different colonies spawning at different times throughout the year. The discovery that spawning was synchronized, that it occurred over just a few nights per year, that it involved virtually every coral colony across hundreds of kilometers, revolutionized our understanding of coral reproduction and reef connectivity. In the decades since, mass spawning has been documented on reefs worldwide, though the timing varies by location. Caribbean reefs spawn in August and September, following different moon phases and temperature cues than Pacific reefs. Red Sea reefs spawn in June and July. Each region has its own schedule, but the fundamental pattern of synchronization remains. Wherever coral reefs exist, corals have evolved mechanisms to coordinate their reproduction, ensuring that the ocean is flooded with gametes all at once, overwhelming predators and maximizing fertilization success. The environmental conditions required for spawning are becoming harder to achieve as oceans warm. Corals stressed by high temperatures often abort spawning, resorbing their gametes rather than releasing them, conserving energy for survival rather than investing in reproduction, with poor chances of success. Bleached corals rarely spawn. Even sublethal stress can reduce fecundity, with stressed corals producing fewer or smaller eggs. This reproductive impairment compounds the direct mortality from bleaching. Even reefs that survive a bleaching event may suffer reduced recruitment for years afterward as stressed colonies fail to reproduce effectively. Yet the spawning continues, year after year, proof that despite all the stresses facing coral reefs, these ancient organisms continue their eternal work of reproduction, continuing to release their gametes into the ocean in the quantities necessary to create the next generation. Every October and November, 120 minutes after sunset, when the moon is full and the water temperature is right, billions of coral colonies on the Great Barrier Reef release trillions of gametes, painting the ocean with color, filling the water with life, betting on the future even as that future becomes increasingly uncertain. The larvae that result from this annual spawning carry within them genetic information, accumulated over millions of years of evolution, blueprints for building reefs that have worked successfully since before the dawn of human civilization. They are hope made visible, floating through the dark water, seeking the substrate where they will begin the next chapter in the endless story of coral reefs. Each larva is a lottery ticket, a chance at survival, a possibility that the reef will persist for another generation. Scientists who witness the spawning describe it as one of the most moving natural phenomena they have ever seen, a reminder that life finds ways to persist, to reproduce, to continue despite all obstacles. The sheer abundance of spawn, the coordination across such vast scales, the ancient mechanisms that make it all work, the beauty of countless millions of pink and orange spheres rising through the water. It is nature at its most magnificent, life asserting itself with overwhelming force. Tomorrow, we will look forward to the future of coral reefs, to the technologies and approaches that might allow these ecosystems to survive the challenges of the 21st century and beyond. We will explore visions of what reefs might become, how human intervention might preserve them, what we might lose if we fail. But tonight, remember the spawning. Remember the sight of 1,000 coral colonies releasing their gametes simultaneously. Remember that these organisms have been reproducing through this same basic process for hundreds of millions of years, that they have survived ice ages and asteroid impacts and every challenge Earth could present. They are survivors. They are extraordinarily resilient. And if we give them a chance, if we act quickly enough to stabilize the conditions they need, they will continue this ancient ritual for millions of years to come, filling the ocean with new generations of reef builders, painting the seas with color, creating paradise through their endless patient work of turning dissolved minerals into living stone. By the year 2100, coral reefs might exist primarily as assisted ecosystems, living structures that persist only through active human intervention and management. This vision of the future troubles some who believe nature should remain wild, unmanaged, free from human interference. But the alternative, the complete loss of coral reefs as functional ecosystems, is more troubling still. The question is not whether humans will intervene in reef systems. We already have, dramatically, through the carbon we have released and the warming we have caused. The question is whether we will intervene to help reefs survive the changes we have set in motion. The technologies being developed or imagined for reef conservation span from proven approaches being scaled up to speculative ideas that may or may not prove feasible. Genetic modification, controversial but potentially powerful, could create corals with enhanced abilities to tolerate heat, acid, or disease. Scientists have already identified specific genes associated with heat tolerance in Persian Gulf corals and other populations that survive in extreme conditions. In theory, these genes could be transferred to corals from other regions, creating heat-resistant strains that could survive in warming oceans where their wild ancestors would perish. The ethical considerations are complex. Would genetically modified corals successfully integrate into reef ecosystems, or might they have unexpected effects on other species, on food webs, on ecological relationships refined over millions of years? Would they be able to reproduce with wild corals, and if so, what would happen to the genetic integrity of wild populations? Would the modification of corals to tolerate human-caused warming be an admission of defeat, an acknowledgement that we will not address the root causes of climate change? Or would it be a pragmatic recognition that we must work with the reality we have created, using every tool available to preserve biodiversity through a period of rapid environmental change? These questions have no definitive answers, but they are being asked by scientists and ethicists because the stakes are so high. If genetic modification could preserve reefs, prevent the extinction of hundreds of coral species, and the collapse of ecosystems supporting millions of other species, would we be justified in not using it? Conversely, if we modify organisms to survive in altered environments, do we lose something essential about wildness, about nature's independence from human control? Beyond genetic modification, other advanced technologies might reshape reef conservation. Probiotics represent a less invasive approach, supplementing corals with beneficial microbes rather than modifying the coral's own genome. Researchers are discovering that coral health depends heavily on their microbiome, the community of bacteria, archaea, fungi, and viruses that live in and on coral tissue. Just as human health depends on maintaining a healthy gut microbiome, coral health depends on maintaining beneficial microbial partners. Some bacterial strains protect corals from diseases. Others help corals tolerate heat stress or recover from bleaching events. Scientists are developing cocktails of beneficial microbes that could be cultured in laboratories and sprayed over reefs, inoculating corals with microbial communities optimized for resilience. The approach is still experimental, but early trials show promise. Corals treated with specific probiotic mixtures show improved survival rates under stress compared to untreated controls. Artificial upwelling represents a physical rather than biological intervention. The technology pumps cold water from depth to the surface, cooling reef areas during heat waves. The concept is simple, but implementation is challenging. The pumps require enormous amounts of energy. The cold water brought from depth is nutrient-rich, which could trigger algal blooms that harm rather than help reefs. The area that can be cooled is limited, making the approach viable only for small, high-value reef sections, rather than entire reef systems. Yet for protecting critical areas, key source reefs that supply larvae to downstream sites, or reefs of exceptional biodiversity, artificial upwelling might provide targeted protection during extreme heat events. Cloud brightening, mentioned earlier, operates on a larger scale, potentially cooling entire reef regions by increasing cloud cover and reflectivity above the ocean. The technique sprays microscopic seawater droplets into the air, creating clouds that reflect more sunlight back to space. Early experiments suggest cooling of 1 to 2 degrees Celsius is achievable, but the approach raises questions about unintended consequences. Would altered cloud patterns affect rainfall on nearby land? Would changes in sunlight reaching the ocean alter phytoplankton communities in ways that cascade through marine food webs? Would the technology be effective enough to justify its cost and complexity? Three-dimensional printing of reef structures offers a more straightforward approach. Calcium carbonate or similar materials can be printed into complex shapes that mimic natural reef architecture, creating substrate that corals colonize much faster than flat surfaces. The printed structures can be designed with optimal complexity, including the caves and overhangs that fish require, the surface textures that encourage coral settlement, the structural strength to withstand storms. Deploying printed reef structures accelerates the physical recovery of degraded reefs, providing the framework that organisms need even before corals have regrown significantly. Looking further ahead, some scientists envision floating coral cities, mobile reef platforms that could be moved to track optimal temperature zones as oceans warm. The concept seems far-fetched, but it addresses a fundamental problem. Reefs are sessile, unable to migrate as their optimal habitat shifts poleward with warming. Coral colonies themselves cannot move, but if entire reef platforms could be relocated, carrying their communities of corals and associated species to new latitudes as needed, reef ecosystems might persist even as tropical zones become too hot for corals to survive. The engineering challenges are immense, the costs astronomical, but for preserving critically endangered coral species or maintaining reefs for their economic and ecological value, the approach might become viable. Even more speculative, some researchers imagine coral farms on Mars. The idea is not as absurd as it might initially sound. Coral calcification removes carbon dioxide from water and produces oxygen as a byproduct, especially when the corals host photosynthetic zooxanthellae. In a closed environment like a Martian habitat, organisms that consume carbon dioxide and produce oxygen, while also creating structural materials from dissolved minerals, could be extraordinarily valuable. The calcium carbonate that corals build could serve as construction material. The oxygen they produce could supplement life support systems. The research being done to understand coral biology for conservation on Earth might someday enable coral-inspired technologies for space colonization. But return to Earth, to the present and near future, to the reefs that exist now and the question of what they will become. The economic value of coral reefs provides powerful motivation for their conservation. $375 billion annually through tourism and fishing. 500 million people depending directly on reefs for food and livelihoods. Coastal protection services valued at over $9 billion per year, as reefs absorb wave energy that would otherwise erode shorelines and damage coastal infrastructure. These are not abstract ecological values, but concrete economic benefits that national governments and international organizations can calculate and plan around. Beyond economics, reefs harbor biochemical treasures of immense pharmaceutical value. Compounds isolated from reef organisms have produced treatments for cancer, including the drug cytarabine derived from Caribbean sponges, which treats leukemia and lymphoma. Antivirals developed from coral reef organisms show promise against HIV and herpes. Painkillers based on cone snail venom are among the most powerful non-addictive analgesics known. The biodiversity of coral reefs represents an unscreened library of potentially useful compounds. Molecules evolved over millions of years to perform specific functions, many of which might be adapted for human medicine. Every coral species that goes extinct takes with it unique genetic information, compounds that exist nowhere else, potential solutions to medical problems we have not yet solved. The preservation of reef biodiversity is, from this perspective, an investment in future human health, a library we would be foolish to burn before we have read even a fraction of its contents. Traditional knowledge systems are being recognized as essential components of reef conservation. Indigenous and local communities have been managing reef resources sustainably for millennia in many cases, long before Western science began studying these ecosystems. The knowledge embedded in traditional practices, in the timing of fishing closures, in the designation of protected areas, in the cultural taboos that regulate resource use, often aligns with and complements scientific understanding. Increasingly, conservation projects are incorporating traditional knowledge, working with communities as partners rather than viewing them as obstacles to conservation. This approach is more likely to succeed because it respects existing relationships between people and reefs, because it draws on deep knowledge of local conditions and patterns, and because communities that have ownership over conservation decisions are more likely to support and enforce protections. The merger of traditional ecological knowledge and modern science creates more robust, culturally appropriate and effective conservation strategies. The future of coral reefs will likely involve all of these approaches and more. Traditional management integrated with modern science, genetic tools used alongside habitat protection, engineering solutions deployed where necessary, but not replacing natural processes, economic valuation, informing policy decisions, and international cooperation on climate change mitigation. No single approach will be sufficient. Reefs face multiple stresses simultaneously: warming, acidification, pollution, overfishing, coastal development, disease. Addressing only one stress while ignoring others will not preserve functional reef ecosystems. Yet perhaps the most important lesson from 500 million years of coral evolution is that reefs are resilient. They have survived mass extinctions that eliminated 96% of marine life. They have persisted through ice ages when sea levels dropped by over 100 meters and tropical zones shrank dramatically. They have recovered from asteroid impacts, from extreme warming events, from ocean acidification episodes far more severe than what we are currently causing. Each time, they took millions of years to fully recover, but they did recover. The genetic toolkit for building reefs, for forming symbioses, for calcifying efficiently, these capabilities have persisted through every catastrophe Earth has experienced since corals first evolved. This resilience gives us reason for hope, but it does not guarantee that reefs will survive the current crisis. Past recoveries occurred over timescales of millions of years, far longer than human civilization will wait for ecosystems to recover. Past recoveries occurred without the additional stresses that modern reefs face from overfishing, pollution, and direct habitat destruction. Past recoveries occurred in an ocean not being fundamentally altered by the activities of a single dominant species. The reefs that exist in 2100, if they exist, will likely be different from the reefs of 2000. They may be dominated by different coral species, the heat-tolerant and fast-growing species that can survive in warmer water and recover quickly from disturbances. They may harbor less diversity, with specialist species replaced by generalists more capable of tolerating variable conditions. They may exist primarily in areas that are actively managed, protected, and restored, rather than existing as truly wild ecosystems. But they could still be reefs. They could still build limestone structures that provide habitat for fish and invertebrates. They could still protect coastlines from storm waves. They could still inspire wonder in people who visit them. They could still evolve, adapting over generations to changing conditions, becoming something new while retaining the essential characteristics that make them coral reefs. The question is not whether reefs will be identical to historical reefs. They will not. The question is whether they will continue to exist as functioning ecosystems that provide ecological and economic services, that harbor significant biodiversity, that demonstrate the creative power of evolution, that connect us to the deep history of life on Earth. Coral reefs teach us lessons that extend far beyond marine biology. They demonstrate how cooperation, the partnership between coral and zooxanthellae, can be more successful than competition. They show how the smallest organisms, polyps barely visible individually, can create structures that endure for millennia and rival human construction in scale and complexity. They prove that life can create abundance from scarcity, building diverse productive ecosystems in nutrient-poor tropical waters through efficient recycling and tight coupling of organisms. They illustrate how biodiversity itself generates further biodiversity, as the structural complexity created by corals enables countless other species to exist. If we allow coral reefs to disappear, we lose more than beautiful underwater scenery or productive fishing grounds. We lose living examples of principles that could inform how we design our own systems, our cities, our economies, our relationships with the environment. We lose connection to an evolutionary history older than forests, older than flowers, older than any terrestrial ecosystem. We lose laboratories where new compounds and organisms are still being discovered at rates of dozens per year. We lose coastal protection that shields hundreds of millions of people from storm damage. We lose food security for communities that depend on reef fish as their primary protein source. The stakes could hardly be higher, yet the tools exist to address the crisis. We know what must be done to stabilize climate. We know how to protect and restore reefs. We know which interventions work and which do not. What remains uncertain is whether humanity will act with the urgency that the situation demands, whether we will treat the coral crisis as the emergency it is, whether we will devote the resources and international cooperation necessary to preserve these irreplaceable ecosystems through the difficult decades ahead. The coral polyps building reefs today, the tiny architects extracting calcium from seawater and depositing it atom by atom as aragonite, are doing the same work their ancestors did 500 million years ago. They are following genetic instructions refined over evolutionary time. They are building paradise beneath the waves, creating structures that will outlast them by centuries, contributing to a project far larger and longer than their individual lives. They know nothing of climate change, of ocean acidification, of the debates humans have about their future. They simply build, day after day, doing what evolution has equipped them to do. Our task is to ensure they can continue that work, that the ocean remains suitable for reef building, that coral reefs remain part of Earth's future as they have been part of its past. The challenge is immense, but so is the value of what we are fighting to preserve. Coral reefs are not expendable. They are treasures, both for their own sake and for everything they provide to the planet and to humanity. They deserve our best efforts, our most innovative thinking, our sustained commitment. They deserve a future, and if we act now with determination and creativity and cooperation, we can give them one.